ठीक है, हो गया।
Kamil sir, you want to say something? Sir, it is 9:05. Shall we start?
Wait, I think Dr. Rowling has to join. He will join within 5 minutes.
Okay, sir.
You check, Bombay ma'am, whether Rowling has joined or not.
No, sir. I don't.
You check
I don't
just.
see him, sir. Coach, sir.
Yes,
Has
ma'am.
Rowling sir joined?
No. Shaolin sir has joined.
Yeah, he has joined.
Yes, sir.
Which name?
Shaolin Singh.
Okay, you make him finish.
Yes, I have met.
Yeah, he has joined. Okay. I think we should continue.
So shall we start?
Yeah, he has joined. You welcome him.
Yes, sir. A very bright and lovely morning to all of you present today. I, Mrs. Memuna Bombewala, coordinator of inaugural session, on behalf of Yatmal Zilla Vikas Samiti's P. Vadwani College of Pharmacy, welcome all our dignitaries present for this 3-day long international webinar series on Pharmacy 360-degree Research to Practice, organized by Patil Vadwani College of Pharmacy in association with APTI, Association of Pharmaceutical Teachers of India, and co-sponsored by Gentech Life Sciences India. P. Vadwani College of Pharmacy Alumni Association welcomes all of you. I heartily welcome Dr. Mahavir Chogle sir, our speaker for today's function, and also welcome Ravleen Singh Khurana sir, who will be inaugurating today's function. I heartily welcome all our dignitaries from industry, faculty present from various colleges all over India and abroad, and all our dear student fraternity on behalf of our esteemed institute in this webinar, which is now live on YouTube and Zoom. Now, I would like to invite Dr. Nitin Kochar sir to give us a brief introduction about the international webinar on Pharmacy 360 Research to Practice. Over to you, Kochar sir.
Thank you, madam. I welcome you all. It's my honor to be a coordinator of this spectacular international webinar series, Pharmacy 360-degree Research to Practice, which is conducted by P. Vadwani College of Pharmacy Yavatmal in association with Association of Pharmaceutical Teachers of India, APTI. As a coordinator of this event, I heartily welcome convener and principal Dr. Anil Chandewar sir, chief guest of this event Dr. Ravling Singh Khurana, who is the director of Nitika Pharma. Today's speaker Dr. Mahavir Chaugule sir, who is an associate professor at University of Mississippi School of Pharmacy, USA. Chairperson of today's session Dr. Pranita Kashyap madam, who is the principal of Dr. R.G. Bhoyer College of Pharmaceutical Education and Research, Varda. Various faculty members from different colleges of India and abroad, in persons from various industries and students of various colleges from different parts of country. I am very happy to declare that we have received very tremendous response for this event, which is witnessed by huge registrations from not only different states of country, but also from abroad. About 1,900 participants have registered and joined this event by Zoom and YouTube Live. This is a 3 days international webinar series. Today on first day, we have Dr. Mahavir Chaugule sir, who is an associate professor at University of Mississippi School of Pharmacy, USA. He will be sharing his knowledge on aerosolized nanomedicine mediated lung cancer therapy. Tomorrow on 15th of July at 4 p.m., we have Dr. Subrata Dev sir, who is an associate professor at Larkin University College of Pharmacy, USA. He will be delivering his talk on pharmacogenomics in drug disposition, the case of COVID-19 investigational drugs. On 16th July at 9 a.m., we have Dr. Prashant Sakharkar sir, who is an associate professor at Roosevelt University College of Pharmacy, USA. He will be delivering his lecture on is the evidence enough, synthesizing and validating evidence for improving health outcomes. We are very happy to, we are very lucky and fortunate to have such great personalities with us. I am thankful to APTI for giving consent to take this event in association with them. I am also thankful to Genetic Life Sciences Private Limited, P. Wadhwani College of Pharmacy Students Alumni Association, and V. Life for extending their hands for making this event successful. I would like to bring to your information that you can join webinar on all 3 days by the same Zoom link and YouTube Live. The link is same for all 3 days.
Coacher sir, kindly unmute yourself.
You will only have to raise the hands for asking the questions. It's compulsory to remain present for all 3 days. On last day, after filling feedback form, you will get the certificate of attendance. So thank you all once again for making this a great successful event. Over to Bombewala madam to proceed further.
Thank you, Coacher sir, for giving us a brief introduction about the seminar and what we are going to achieve by considering this web series. Now I request Kate sir to introduce Ravleen Singh Khurana sir to us. Kate sir.
Thank you so much, Bimala Bombela madam. Am I audible?
Yes, sir, you are.
Very good morning, all of you. I am Milind Kate, lecturer, P. Wadhwani College of Pharmacy, Yavatmal. It is a great privilege to me on behalf of P. Wadhwani College of Pharmacy, Yavatmal to introduce today's our special guest for inauguration, Mr. Ravling Singh Khurana. Mr. Ravling Singh Khurana, born in 1971, is an Indian excipient manufacturer named Nitika Pharma. He is the developer of many unique processes and first in Asia to fill US DMF for magnesium stearate in the year of 2003. In 1991, Mr. Ravling Singh Khurana started his business with a humble beginning as a young entrepreneur, inspired by then managing director of Ranbaxy at the age of mere 19. He took the decision to start his own company. Today, Nitika Pharma is one of the world leading excipient manufacturer company, exporting to more than 19 countries, catering almost all major global MNCs. From a single digit turnover, the company has grown in many folds. Today, with the turnover of about 100 crore per annum, he has made technical excipient presentation in various forums like Europe, Middle East, Africa, China, and India. He is awarded and recognized by National Award for Outstanding Entrepreneur 2016, Leading Excipient Company by UBM 2015, Most Promising Brand in Pharmaceutical Excipient 2016, Star Export House Award 2016, Leading SME of India Award 2014, Best SME in Pharma Sector Business Gaurav Award 2012. He is also honored to be a part of many associations. President of Laghu Udyog Bharti Nagpur, Post Secretary of VDMA, Secretary of LOC UDP Alumni Association Nagpur. He has ex-member of board of Studies Faculty of Medicine Nagpur University, Chairman with CSR Committee Rotary Club of Nagpur. This is short about Ravling Singh Khurana sir. Now I would like to hand over the session to Memina Bombewala madam. Over to Memina Bombewala.
Thank you, sir. We are so privileged to have such an inspiring personality like like Mr. Ravling Singh Khurana sir with us today. Now, I would like to request Mr. Ravling Singh Khurana sir to give us his precious guidance.
Hello.
Yes, sir.
Yeah, I'm audible to all, right?
Yes, sir.
Yes, sir.
Yeah. Good morning, everyone.
Morning.
I am thankful to Dr. Chandevar and all the students of Advani College of Pharmacy for inviting me to this web series. I am also a student of Advani College of Pharmacy. I have submitted my PhD from this college and I am still waiting for it to be awarded. So when Bombewala madam called me Dr. Khurana, I said I am waiting. अभी वो बाकी अमरावती यूनिवर्सिटी का काम थोड़ा सा हो जाए. So maybe 2017 I have submitted my PhD and hopefully this year it should be done. So it's great to be amongst you students. Pharma has been my passion right from I was student. I joined University Department of Pharmaceutical Sciences in the year 1988. And at that time, pharmacy was something not very great as it is today. And people even used not to know what bee farm is all about. So bee farm, they used to think is something to do with the farming. Farm यानी farming आप करोगे. Otherwise, medical dukan, that's the what people thought about it. But as last 30 years, the profession has grown very nicely in India and abroad. And today, the situation is, I have traveled more than 60 countries, and everywhere you'll find India is a great name in pharma research, in pharma manufacturing, and the whole world is looking upon India to deliver the quality pharmaceuticals to the whole world. Recently, the corona epidemic has further strengthened Indian pharmaceutical industry with President Trump, President of USA, Mr. Trump, asking for hydroxychloroquine tablets from India. So that shows the Britishers, the US people, and even the Brazilian Prime Minister requested our Prime Minister Modi to give him some paracetamol tablets in bulk because they are facing shortage of paracetamol. So truly, we are the pharmacy of the world. And when we were also especially happy when the hydroxychloroquine was delivered to USA by IPCA, because Nitika was supplying the key excipient magnesium stearate to these tablets which were supplied to the US. So pharmacy is a great profession. And whenever world faces such pandemics, people realize the importance of pharmacy and pharmacists. Today, people are talking more about doctors, nurses. Yes, they are in the forefront. But as the epidemic is progressing, everybody is looking up to the pharmaceutical companies to develop new drugs, to develop vaccines, and save the world. So ultimately, it's pharmacists and the pharmaceutical industry who will save the world from this epidemics. So in India, the pharmacy is pharmacies education started somewhere in 1946 with BHU was one of the oldest college, and then started colleges of Chandigarh, Bombay University, UDCT, and Nagpur University. So the Nagpur University College UDPS, which could be a mother college for all of you, because Dr. Chandewar was my colleague, my senior colleague in the UDPS, and many other faculty members all over Vidarbha are coming from this college. They will appreciate that this college has produced so many stalwarts that we are proud to have the owner of Ajanta Pharma, which is among top 5 companies in India, from this college. Then also we have the Drug Controller General of India, Dr. Venugopal Somani, is an alumni of our college. He was my few batch senior. So he is today taking the decisions. And I'll also mention here that Favipiravir, which has been recently launched by Glenmark, it was the extraordinary efforts from Dr. Somani that he approved this drug with a 3 months of shelf life. So it's rare in the history of India that a drug is being launched by this special mechanism where Dr. Somani worked day and night. He worked sometime 2 o'clock till night to ensure that this drug is on the shelves, and today many lives are being saved because of this drug. Similarly, there are many other stalwarts like Dr. Kokate, who was the president of Indian Pharma Congress and also held many positions in Government of India. And today there's a committee made, which is called Kokate Committee, which is going to decide the rational formulations all over India. So there are many irrational formulations in the market. And now Kokate Committee, who is reporting to the Prime Minister's office, is going to decide on the formulations that are going to be in the market in the form of formulations. So I mean to say that we pharmacists, maybe coming from UDPS, it's the old college, or from other colleges, are doing a great job. And many a times, common public doesn't know about what we are doing. But that should not deter us, or that should not prevent us from doing the great job or great profession that we are into. Drugs have been always an integral part of the humans, right from the prehistorical times till now. And the pharmaceutical industry in India is making best drugs in the world. Today, the maximum number of US FDA plants outside USA are in India. And not only that, but 30% of the generic formulations in USA are coming from our country. When I travel these 60, 65 countries, I go to a chemist shop everywhere and try to see like what is there in medicines and what they are making. There will not be a pharmacy, my friends, you'll be happy to know, throughout the world, which will not have Indian drugs on their shelf. So that is the greatness of us Indians. And formulations is, India has a wide range of formulation industry, right from the oral solids like tablet capsules to ophthalmics to injectables to the biologics and the vaccines also. You'll happy to know India is a leader in the vaccine manufacturing in the world. So the highest capacity of vaccine in the world is coming from India. And this is one area where China is also lagging far behind. So I had been to China many times, and what Chinese are doing, they're trying to get Indian scientists there who can train Chinese into the pharmaceutical industry. And they know that the kind of formulations Indians can make, China cannot make it. So there's also one area we are among the pioneer in the world, and we have left China behind. So the today's topic, pharmacy from research to practice, is very relevant. Why? If you don't do research and if you just focus on manufacturing, what will land up into mostly is a Me Too drugs, is a formulation like somebody is making, you're copying and making it. But that's not enough. In today's world, you need to be innovative in everything you do. And my friends, research is not as difficult as you might perceive. There are many ways by your research can contribute to making good quality drugs. like I'll give one very simple example. We make excipients, and excipients are an integral part of any formulation. Many times the excipients are looked down, like excipient ka kya role hai? They are inert materials. But the research has proved, and last week I delivered a webinar, international webinar by the International Pharmaceutical Excipient Council, and emphasizing the role of excipients. So even an excipient, if done or made with the proper research, can ensure your formulation will do its job properly in the human body. And sometimes if the excipient is not proper, your whole formulation could go wrong. It will not deliver the therapeutic effect that you want. So there's a lot of scope of research in every sphere. Even in formulation development, there are many areas where you can have sustained release drugs, where you can have time release options, where you can research on many different facets of industry and add value. Even pharmaceutical packing is a very innovative area, because kind of complex norms are coming up for the packing, and the complex products are coming, they require complex packing solutions. So many pharmaceutical industries are looking for innovative packing materials, and so on and so forth. Even research is required in the pharma marketing. So pharma marketing, you know, is one of the most challenging marketing in the world, and that also requires a lot of research to be done before, market research to be done before you can launch a product in the market or you can make it successful. So whenever you are doing or backing something with the research, you have that advantage. So anybody who is doing something for the first time will definitely have a lot of benefit over others. Others may copy it, but original remains original. Like even like there are many different brands will keep on coming. But if you made an innovative formulation, and there are some companies, even small companies, who are developing very innovative formulations, and they are doing very well. I go to Baddi also, which is today the hub of pharma industry in India. And many people say, कि अरे Baddi में क्या research होता है, वो तो खाली copy करते हैं. But there are some small industries in Baddi which started as a very small, and today they have reached in among the top industries of the country. So I would like to again emphasize on this, that research plays a very important role, and we all should focus on doing the quality research and the research which is relevant to the industry. If we carry out some research which is not going to translate into the reality, or it's not going to see the light of the day, that research might not have a lot of value. It will remain in books. So also it's important to do research which is going to contribute something to the well-being of the patients, which is our ultimate aim. So my friends, I'll not take much of your time, and I'll once again encourage you to take research and start from research to the practice of pharma, which includes manufacturing, which includes marketing and practice. So all facets of pharma industry, starting from research to manufacturing to distribution to marketing, are equally important. And I'm sure you all will prosper in the different areas of pharmacy and make the profession proud of you. Thank you very much.
Thank you, sir. The glow of fun, warm thought is more precious than many things in the world. And such were your words of inspiration, sir. You have again made us proud of being a pharmacist and motivated us to work in and for our field. And that should not be for namesake. The beginning is the most important part of the work. Or saying it even better would be saying that making the beginning is 1/3 of the work done. So I would like to invite Mr. Ravling Singh Khurana sir to inaugurate today's event by virtually cutting the ribbon and lighting of the pious lamp. Thank you, sir.
Thank you, madam. I think some screen is being shared, right?
Yes, sir. There is a screen share.
Okay.
We just have to bear with few seconds.
No problem. Yes, it's there.
Everyone has to clap.
Corona has taught us how to live with the new normal, and we are very adaptive people. We have adopted things very fast. I'm very happy the way you all are conducting this webinar. So very nice approach, Chandevarji. Keep it up. My best wishes to the web series.
Thank you, sir.
There is one more screen share.
शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः.
So Vadhwani College students have given an example of how innovative they are. They make the lamp lit, EE lamp lighting. They have done very nicely. Very innovative.
Thank
Thank
you,
you,
sir.
Rauli.
Thank you so much.
Welcome.
I would now request our team head, mentor, guide, and inspiration, Dr. A.V. Chandevar, sir, Principal P. Vadhwani College of Pharmacy, to address our dignitaries.
Okay. Good morning to everybody.
Good morning.
I welcome all of you for this webinar, the Pharmacy 360-degree Research to Practice. This is your first day. First of all, I am very much thankful to Dr. Rauli. Yes, I can say him doctor because he don't need any type of research because he's already achieved that one. As in the introduction, it has been mentioned that he has traveled almost the world. And we are very proud that he is from alumni of the Department of Pharmaceutical Sciences. He is my immediate junior. He is a good friend of mine. And he is very always kind, very supportive to us whenever we ask his help. He is always ready to do that one. So thank you, Raulin. Within a short notice, even you accepted my invitation and agreed to inaugurate our function. And because of your presence, this is becoming very attractive one, I can say, and very informative also. I am again thankful to today's speaker, Dr. Mahavir Chaugule. What to say about him, Dr. Mahavir? Dr. Mahavir is my student. He's a student of our college. He's our alumni, and he is very our proud alumni. He's a student of the first batch of our college. So when he was in our college, he was very studious, very sincere, and very calm and quiet. And he developed himself. He got postgraduated, and he went to USA, and now he is in the teaching profession there and doing well. I am getting various information about him that he is very well. He's a very informative person, very knowledgeable person. So I am thankful to Mahavir also. Even with a short notice, he also accepted our invitation and agreed to be a speaker during this webinar. I'm thankful to all the participants, the college, various participant faculty members who have been participating here. I'm very much thankful to Dr. Pranita Kashyap ma'am, who accepted our invitation to be the chairperson for today's webinar. Dr. Maria and all principals from various colleges, professors, associate professor, assistant professors, the persons who are joining from pharma industry, and also our student from research and postgraduate. I welcome one and all for this webinar. Dear participants and dear guests, we have taken the opportunities and the advantage of this COVID-19 situation. Whenever our apex bodies have asked us to go for various webinars, seminars, and online classes, we've taken the opportunity and we've taken the advantage of that. And first, we started with the very short with 1 day seminar, that is blended teaching learning process. So that was the very was the informative one. So how to use the various informative tools in the teaching learning process. So that was the first seminars we have conducted online, and later on the series of various webinars we have conducted. We have conducted 1 week faculty development program where various eminent persons they have delivered their lectures. Dr. Sital Kumar Zhamwar, Dr. Veena Belgamwar, Dr. C.V. Pardesi, Dr. R.D. Thakkar, Dr. Anwar Sheikh, Professor Vandana B. Patel, and Professor Dr. Satish Pulsar. Very eminent persons, they have delivered, they have delivered their knowledge, they have given the ideas about the pharma industry, and the topic were also the very innovative, and it's a huge response for this faculty development program. More than 1,000 people have participated, and that is the one first kind of it's in Maharashtra, I can say, or all over India, that we have taken E type of faculty development program. Later on, various faculty development program came out, but that was the first. You can say that in the 5th May to 9th of May 2020, we have conducted that one. Then the continuous series of webinars we have conducted, where 1 day online national workshop was conducted on 17th May, the modern teaching technique using informative and communication technology, where our supporter, you can say, V Life, Mr. Abhijit, and our professor, Mr. Sheikh, have delivered lecture on that, how to utilize the modern tools in the teaching learning process. Then 1 day national level webinar on ICS guideline pre and post COVID was organized on 20th May, where Dr. Rana Zainuddin, HOD, YB Chavan College of Pharmacy, delivered a lecture. Then we have conducted a 2 days national webinar, that is current scenario in psychoneuropharmacology. In that, various eminent persons have participated from IITs and NIPER. Dr. Bhupesh Sharma, professor, Amity Institute of Pharmacy, Amity University, Uttar Pradesh, delivered seminar. Then Dr. Manoj Dandekar, who is a professor, head pharmacology and toxicology, Department of National Institute of Pharmaceutical Education and Research, NIPER, Hyderabad. He has delivered lecture on mapping of depression switches. Then Dr. Divya Vora, professor and head Department of Pharmacology, she has delivered lecture on a current experimental and translation approaches for anti-seizures and anti-epileptic therapeutics. Then Dr. A.K. Thakur, professor, biological sciences and bioengineering department in Indian Institute of Technology, IIT Kanpur, has delivered seminar on peptide model to understand mechanism of polyglutamine aggregations and targets for drug delivery for hospitals and diseases. So these eminent persons have delivered the lecture, and one more, that is the a short-term training course we have conducted. In that short-term training course, that was the 1 week that was conducted, and it was also very, you can say that success in that various participants have participated in that short-term training course. As far as dear participants, our college journey, we have to say, the college has started, I just summarize in short because we're very late. The college was started in 1997, and continuously with the help of our management, our colleagues, staff, I can say the very energetic faculty I am having. So with the entire efforts of our faculty management, we achieved a lot in a short span. I can just summarize here the achievement of the college, that we are the first in pharmacy colleges in Amravati region, having the 2F and 12B UGC recognition, that the permanent affiliation from the Amravati University. We have certified ISO 9001 and 2015 college. Then we have received various grants from AICT. Recently, we received the grant of 17 lakhs to Professor Chandawar and Modroffs. Previously, we received the grants from AICT under the RPS scheme, Modroff scheme, and various grants for conduction of the seminars also. We are having the PhD centers in Amravati region, and the very maximum number of seats we are having. 32 seats we are having for the PhD research activity. More than 500 people have been published by our college. This faculty from our college, very reputed journals, Science Direct, Engenta Connects, Springer Link, etc. And around 12 patents have been awarded to the faculty. Number of books coming out, various books have been written by our faculty members. We received award for the best, this you can say, for the Swachhata Abhiyan, Maharashtra Swachhata Abhiyan survey. So we were the second prize winner in the Yavatmal district. Then our faculty have traveled abroad also for presentation their research paper. Then our students are participating in various sports activities. Mr. A.K. Mesra has received the color code in the table tennis. Our institute has received the gold rank in 2019 in the AICT C2 survey. And presently, we have been ranked in between 76 to 100 band by the National Institute of Ranking Framework, Government of India, MHRD. So that is our big achievement, that we are the first in Amravati region, and we are the second in Vidarbha region. I should mention here the Sadashiva Patil College of Pharmacy, Kamti is the first one here. They have ranked very highest rank in 40th, and we are the second for placing in the 100 band by NIRF. Our college has been shortlisted for Utkrishta Sansthan Vishwakarma Award 2019, which is also the very big achievement for us, because we were the first college in Maharashtra to be shortlisted for this award. So this is our short achievement. Recently, we are going for the NAC accreditation. And as far as this webinar is concerned, I must thankful to President of APTI, Praveen Chaudhary, who has accepted to be associated with us to sponsor this webinar. I must be thankful to Mahavir and Mr. Dev, and specifically I should be thankful to Professor Sakharkar, who is a great and good friend of mine. He has started his career from IPR Varda, and he was colleague there with me. So his suggestion was there to conduct the international webinar, and he accepted to arrange all the speakers. And he spoke with all these two speakers, and then this webinar series came out. So once again, I welcome all of you. Once again, I'm thankful to Pravleen, Dr. Mahavir, and all the participants, Professor Kashyap, Professor Madia, Elsie Abraham, I see she has also joined this webinar, and all my colleagues from teaching people. I'm very much thankful to our alumni association, that is PWCOPSA, Association of Pharmacy Student Alumni Association, for being the associated with us for this particular seminar. I'm also thankful to Genetic Pharma, particularly Dr. Mahendra Kshirsagar, who has accepted to be associated with us for this webinar. So once again, welcome all of you. And I hope that as the beginning is very successful, I hope that this entire series of the webinar very, very successful, very important. And giving the best of luck. Not at the last, not in the least, I must be thankful to our beloved chairman, Jagdish Wadhwani sir, who is always very supportive to us. And when I have given the idea about the webinar, he also have given us the green signal that is continue. And he's a patron for this particular webinar. So I'm thankful to him, and I'm thankful to management person. So once again, best of luck for the 3 days webinar series. Thank you. Over to Bombay ma'am.
Thank you, sir. With this, we are coming towards the end of the inaugural ceremony. So I request Mr. Kate, sir, to present a vote of thanks.
You just give that immediately after that the first session will start.
Yes, sir.
Thank you so much, Bombay madam. Once again, I am Milind Kate. Honorable Principal Professor Dr. A.V. Chandeva, sir, and today's Chairperson Dr. Kashyap, respected Speaker Mahavir Chaugule, sir, Mr. Ravlin Singh Khurana, sir, and all the participants. It is my privilege to have been asked to propose an inauguration vote of thanks on this occasion. I, on behalf of P. Advani College of Pharmacy, Vatva, I would like to thank today's Chairperson, Dr. Kashyap. I extend and really heartily thank to our today's eminent speaker, Dr. Mahavir Chaugule, sir. I would like to thank our beloved principal and convener, Professor Dr. A.V. Chandeva, sir. I would like to grateful thanks to this our special guest, Mr. Ravlin Singh Khurana, sir. I would like to sincere thanks our coordinator, Dr. Nitin Koche, sir, and associate coordinator, Dr. Abhijit Srirao, sir. I would like to thanks today's sessional coordinator, Ms. M.D. Kitukle, madam, and Dr. S.R. Gawande, madam. Last but not least, a big thanks to our participate in today's inauguration. Let's enjoy next 2 days this international webinar on pharmacy 360 research to practice. Now I concluded that the inauguration session is over. Now I would like to hand over the session once again to Memuna Bombewala, madam. Over to Bombewala, madam.
Thank you, Kate, sir. With this, I declare that the inaugural function is over and will be soon starting with our plenary session 1 today. I would like to hand over the mic to my friend and colleague, Dr. Shilpa Gawande, who is the session coordinator for webinar 1 on the topic aerosolized nanomedicine mediated lung cancer therapy. Madam, over to you.
Thank you, Memuna ma'am. Good
Thank you.
morning, all of you. On behalf of P. Vadhvani College of Pharmacy, Dr. Shilpa Gawande, session coordinator for today's session, welcomes all of you in the international webinar series, Pharmacy 360 Research to Practice. And today I feel very proud to announce that the speaker for today's session, Dr. Mahavir Chaugule, is alumni of our college, and he is amongst one of the most bright student of mine. I welcome Dr. Mahavir Chaugule, and his topic is aerosolized nanomedicine mediated lung cancer therapy. And to introduce such an eminent speaker, I like to request Miss M.D. Kitukle, madam. Over to you, ma'am. Kitukle, ma'am.
हेलो हाँ मैं निकलता हूँ ठीक है
Kitukle, ma'am.
A very good morning. Am I audible?
Yes, ma'am.
Very good morning, respected principal sir. Today's guest, Dr. Ravlin Khurana sir. Today's chairperson, Dr. Kashyap ma'am, and speaker of the day, Dr. Mahavir Chaugule, and all dear participants. I feel elated to introduce today's speaker to everyone. Dr. Mahavir B. Khurana. He did his postdoctoral fellow in cancer therapy and nanotechnology, focused on treatment of lung and breast cancer. He's awarded with the Doctor of Philosophy in Pharmaceutical Sciences from Maharaja Sayajirao University, Baroda, and worked on treatment of cystic fibrosis and AIDS-associated pneumonia. He is graduated with gold medal from Amravati University, and we are very proud that he is alumni of our college. And he is known for his work on treatment of lung and breast cancer. About professional experiences, uh, he has a very huge professional experience in a small age. Uh, Presently, he is working as associate professor in Mississippi University, and he has huge experience as assistant professor for the students of PharmD and PhD students. His research focus on targeted pulmonary delivery of drug and siRNA for treatment of lung cancer and asthma. Multi-target based combination therapies for cancer. He's attracted extramural funding of around $410,000 and $715,000 from NIH and NSF. In, he has 25 research publications for his credit, 2 review articles and 7 book chapters and 2 editorial articles in his credit. He has delivered 13 invited talks and presented around 64 posters at national and international conferences. His publications has received more than 600 citations with an H index of 13. Then he's also worked on development and patents for targeted and inhalable oral, parenteral, bioresponsive nanocarriers, a platform technology for drug, protein and siRNA delivery for cancer and asthma therapy. His experience in targeted drug delivery system and molecular biology, NP discovery, and pharmacokinetic, pharmacodynamic, and imaging techniques. Directed and supervised 4 postdoc students, 3 PhD students, and 7 PharmD students under his guidance. He served on several college and university committees and faculty research committees and recommended as a best new faculty member for the recruitment. He attended around 16 teaching research and industrial workshops and received he's a receiver of our best student award of our college. Again, about professional experience, he's also having teaching experience. Currently, he's working on 5 projects based on cancer therapy. Also, he has taken industrial consultancy like Sun Pharma and Tamir Biotech. So we can say we have multidimensional talented teacher presently giving services as associate professor to the Mississippi University. So we are very lucky to have you, sir. So you are inspiration to all the young teachers who are entered in this noble profession. So being inspiration and multidimensional, we are very excited to hear from you, sir. So over to Gavande madam for the further process.
Thank you, madam. Now, for today's session 1, I would like to request respected Dr. Pranita Kashyap ma'am, principal of Dr. R.G. Bhuyer Institute of Education and Research, Varda, to be the chairperson for the today's session. I request ma'am, please ma'am, and also I request Dr. Mahavir Chaugule to start his session.
Thank you, ma'am.
Dr. Mahavir Chaugule, please start your session.
Yes, I just wanted to get my slides on.
Personally, Dr. Mahavir.
Yes.
Thank you for giving a lecture on this webinar series. Thank you very much for accepting our invitation. So thank you very much to you.
Yeah. Thank you, Dr. Chandivar sir, for this opportunity. And always I can say that the source of inspiration, during my college days started from you, as well as, Dr. Gavande madam. And I thank you for a nice introduction. Even the curriculum madam summarized the accomplishments. Thank you very much for that. And it is a proudful moment for me to basically deliver in a seminar based on my research work what we are conducting here associated with the Wadhwani College of Pharmacy. I do know that in the past, Dr. Chandivar sir did ask me to come over to the Vadvani College of Pharmacy, but that didn't happen. But this way, I'm reaching out to my college, so that's a very proudful moment for me. In today's talk, I'm going to talk about the targeted nanomedicines for treatment of lung cancer. So, in the lung cancer, knowing it is one of the leading cause of cancer death in USA as well as worldwide. If you look at the statistics, the lung cancer is number 2 in males as well as number 2 in females. So these are the new estimated cancer cases in 2020 in the US itself. And interestingly, if you see from 1975, the 5-year survival rate is basically unchanged, is the 16%. So even with all the advancement, we have challenges in the lung cancer. So with the lung cancer, if it is surgically removed, that is the first option, surgery. The doctor or surgeon, removes the tumor mass, followed by chemotherapy, radiation therapy, and also recently started immunotherapy. As you may be knowing, most of the cancers, they are very heterogeneous, same as lung cancer has a wide heterogeneity. So with this heterogeneity, basically, the lung cancer develops a resistance to the current treatments, and it will be difficult to treat that cancer. It stops basically responding to the current therapies. So in terms of therapy wise, after surgery, the chemotherapy and immunotherapy is most widely followed approach. Some cases it's effective, but most of the time, you will see severe adverse effects, in the cancer patient. So there is a unmet need to develop newer therapies for treatment of lung cancer, and that's what today's research seminar focused on. So my research is focused on drug discovery and development interface. You may be knowing a lot of investment needs to be made in order to get one drug from lab to the clinic. And with therapeutic agents, you might be aware about small molecule inhibitors, but recently, protein peptides, sRNA and DNA, these are also biomolecules. Those are helpful to treat various diseases. So once we identify the therapeutic agent, most of the time, we do the in vitro basically assays to evaluate the efficacy of specific compound or drug, or even delivery system. In this case, we use different types of methods, like a cell culture method or co-culture method. So we have and we look at the protein expression by using Western blotting to know what are the proteins are underexpressed or overexpressed, and also to evaluate some of the toxicity assays and how overall the mechanistically the drug or nanomedicine works. So once we evaluate under the in vitro efficacy assays, so we go into the pharmacokinetic and pharmacodynamic studies. Most of the time, as you can see, most of the drug end up with having the poor efficacy and higher toxicity. So this can be due to various reasons, maybe a physiological or physicochemical properties of the drug, or limitation of that specific drug, such as like a short half-life, or in vivo degradation, and so on. So our research, we wanted to repurpose the existing drugs or the separate drugs by overcoming the challenges associated with delivery or use of those compounds or drugs, and overcoming some of the limitations. So with the lung cancer, as you know, it is the most common site for the primary malignancy as well as metastasis. So any cancer, you take prostate cancer, breast cancer, when they metastasize, from the blood, they go to the lung, and then lungs, they found a secondary tumors, and those are definitely difficult to treat. So in the lung cancer, there are two types of lung cancer. One is a small cell lung cancer, and the another is non-small cell lung cancer. So the non-small cell lung cancer comprises about 70 to 80% of newly diagnosed cases, and that's where our research is focused on. So in the non-small cell lung cancer, we have a squamous cell carcinoma. As you can see in this figure, the tumor grows near the bronchi. So these are bronchi, bronchioles, mainly bronchioles. in case of large cell carcinoma, the tumor grows near bronchi as well as in the mid region of the lung. And in case of the adenocarcinomas, as you can see, the tumor in this picture, or this in the lung section, it is growing in the peripheral region of the lung. So that is that is the most challenging to treat, and that's where our research we are focusing on. So why we selected the inhalation drug delivery for delivering anti-cancer drug? As you may know, localized drug delivery in the lungs it basically provide us an opportunity to use a lower dose. It minimizes the distribution to the other organs in the body because we are just delivering to the lung, not with other organ. If you look at the conventional delivery systems like oral or parenteral, in that case the drug will go everywhere in the body, all the tissues, and that's where we see the side effects coming from. So we can provide specific delivery, as well as it can be used for local as well as systemic delivery. As you can see, the insulin dry powder inhaler is available in the market for the treatment of diabetes mellitus. It is basically a systemic delivery via inhalation route of administration. And in the lung, it has humongous surface area and low enzymatic activity. So it is very suitable to deliver like sRNA, peptides. Those are we are not able to deliver it by oral route of administration. So in our research, we selected the nebulizers because those are the easiest devices which will help to test the developed formulations. And then they also found to have the deep lung delivery as well as the high respirable fraction. That is the amount of drug which reaches in the deep lung. If you look at the lung deposition, as you can see in this figure, we have inhalation device. The patient inhales it, and it delivers drug to the lung. So this delivery system, or the delivery of the drug to the lung, is highly dependent on the particle size. So if you have particles or droplets of more than 10 micron, so they will primarily deposit into the throat area. If you have about 10 micron or so, they can go in the bronchi. And in order to get the deep lung delivery, so we need a particle size or droplet size of this aerosol cloud between 1 to 5 micron size. So this is the range is needed to deliver any drug to the deep lung. What we did, we observed the COX-2 or cyclooxygenase 2 enzyme that was overexpressed in non-small cell lung cancer. This was we found in our lab. So we were looking for the drug which can inhibit this COX-2 enzyme, and some of you might have took this drug also, the celecoxib, which is a lipophilic COX-2 inhibitor, and it has wide therapeutic index, so very minimum side effects. So, and in the cancer, as we know, we have to use the combination therapy in the cancer area. Single therapy is not going to work because of the heterogeneity of the tumor. So our goal was to use an investigational drug, in this case the celecoxib, which is basically has a wider therapeutic index and also has a minimum side effects, and to combine that one with the FDA-approved drug. Docetaxel is approved for the non-small cell lung cancer. It is given by intravenous route of administration, and this strategy we use in order to evaluate the extent of anti-cancer effect which has occurred due to this celecoxib drug. It is a small molecular weight drug. It has a pK of 11 and oral bioavailability around 40%. One of the limitation of this drug for the oral delivery is it has a first pass metabolism. That means once the drug get absorbed going to the liver, it most of the drug will get degraded in the liver. So with that, the hypothesis we tested is basically if we have an inhalation delivery of this celecoxib solution, and then combine that one with the docetaxel, we will exert a significant anti-cancer activity against the lung cancer. So we used, for this purpose, two cell lines. Actually, one is A549 cells, and another is H460 cells. So these are adenocarcinoma cells. As you can see, these are some pictures of low density and high density of these cells. We got the cells from ADTC. So the first thing, we wanted to know how this silica is interacting with the docetaxel, like whether it is additive effect or whether it's a synergistic effect or it is an antagonistic effect, because the combination of two drugs not necessarily all the time, gives favorable results. So what we did, we selected two concentration of drug, silica solution at 6 microgram per ml, and then 12 microgram per ml. We combined that one with the docetaxel, and we did plotted isobolograms from our data, and also calculated the combination index. So as you can see in this figure, we have silica IC50 equivalent on the x-axis, and then the docetaxel IC50 equivalent on the y-axis. This line is of additivity, so anything or any point below this line is a synergistic effect. Anything we see the point above this line will be a antagonistic effect. So with the combination, we found that it has a moderate synergism to synergistic effect. And this is the combination index scale we have. So we also found that the combination was synergistic in nature. So the quickly what we did is, we wanted to see, our hypothesis, whether it's true, or we wanted to test the hypothesis. So what we did, we used an orthotopic tumor model. So what does that mean? We took 1 million cells of A549 and injected into the lung parenchyma of the mice, waited for 7 days, and that, basically produces the tumors in the mice. And after that, we grouped them in a different group, started the treatment. We continued the treatment for 32 days post-tumor implantation. And after 32 days, we sacrificed the mice, and we removed the lungs uh, and calculated the tumor volume results. So for the delivery of this silica, the solution by inhalation, we also use for the nanoparticle also. So it's an unexposed nose-only exposure chamber. As you can see in this figure, we have a nebulizer here. We put either solution or nano suspension in the nebulizer. Nebulizer is attached to the compressor, which provides the air and then creates an aerosol cloud, and it goes into this unexposed chamber. As you can see, these are the mice holders. We have 6 on the top one and level, and another 6 on the down level. So we nebulize them, for 30 minutes. So in this case, what we did, we had a control group with a placebo aerosol. That means, we don't have any drug in it. Then we had a aerosolized silica solution at a dose of 4.56 mg per kg per day. We also took, oral silica as a control, and then we had a, docetaxel IV intravenously administered at 10 mg per kg. This dose is selected based on its clinical regimen or recommended dose. And then, we combine the aerosolized silica solution with the docetaxel, and then oral silica solution with the docetaxel. So, these are some tumor pictures, but the most interesting results is the tumor volume data. If you see in this figure, we have tumor volume on the y-axis and different treatment group on x-axis. As you can see here, the docetaxel intravenously given did show significant anti-cancer activity. These are, P as a 0.01 level. And then when we combine, the oral silica or aerosolized silica with the docetaxel, did show significant anti-cancer activity, which is at some extent it was supporting the hypothesis that the combination of silica and docetaxel is a synergistic in nature. But surprisingly, the aerosolized silica solution did not show any significant anti-cancer activity, which was not supporting our hypothesis. And also same thing we observed with oral silica. So this led us to a question like, what is the possible reason behind we are not seeing an anti-cancer activity or significant anti-cancer activity with aerosolized silica solution, although we are delivering it by inhalation delivery directly to the lung. So hypothesis should be, if you have a drug giving to the lung, it will show better efficacy. So in order to investigate that, what we did, we conducted the lung deposition studies with this aerosolized silica solution. Again, we use the same in-exposed nose-only exposure chamber. So we aerosolize the silica of the solution at this specific dose. We nebulize, this formulation for 30 minutes, and at each time interval, we collected the blood and isolated the lungs, from 0 to 24 hour, and then we analyze the drug content in all this tissue as well as in the plasma. So if you look at the pulmonary deposition of silica solution, so this is the y-axis. We have percentage dose of silica deposited for lung tissue, or on x-axis we have time. So as you can see in this data, most of the drug is getting cleared within first 2 hours. So after 2 hours, we see a very decline in the drug concentrations. We found similar findings with the plasma levels, as we have the plasma drug concentration versus time, and we evaluated these some of the parameters which I will not go into detail, but the one key observation we observed is the silica is getting cleared rapidly from the lung, and that may be the possible reason why we are not seeing the significant anti-cancer activity. So with that, we wanted to increase the lung residence time of the drug, in this case silica, so that we can achieve the significant anti-cancer effect with this our aerosolized silica system. So when we are looking at, we wanted to increase the lung residence time. So obviously we need to have a system which has a control release property. So, and the silica is lipophilic in nature. So we are looking at the lipid nanocarriers. So for this investigation, we selected a nanostructured lipid carrier. Those are comprised of liquid and solid state lipids in a matrix. It has advantage of having higher drug load capacity and better storage properties compared to a solid lipid nanoparticle, where we have a solid state lipid matrix system. So we selected the nanostructured lipid nanocarriers for this one. So with that, our hypothesis is the inhalation delivery of silica loaded nanocarriers will enhance the lung residence time of the drug, and when we combine that one with the IV docetaxel, will exert in a significant anti-cancer activity. So to test the hypothesis, we have aim one to evaluate this delivery system, whether we are able to deliver it by deep lung and also increase the lung residence time. And then aim two is evaluate, the in vivo efficacy and safety of this formulation alone and in combination with docetaxel. So coming to the formulation aspect, so we use an emulsification process where we had an aqueous phase and oil phase. So the aqueous phase comprised of water, sodium taurocholate is a surfactant, and we heated it, to 60 degrees Celsius. So in the oil phase, we had a completol, which is a solid state lipid, and then we use a magliol, which is a liquid state lipid in this case, and added a drug and added a chloroform to enhance the solubility of the drug. So we homogenize it by using ultratorix in a mixer. We got a coarse emulsion. We took this coarse emulsion, passed through the emulsiplex C5, which is a high pressure homogenizer, for 8 cycles at 10,000 psi, and that gives us silica nanostructured lipid nanocarrier of 210 to 230 nanometer. So the next, we characterize these developed nanocarriers for the particle size and zeta potential. As you can see, the particle size is around 200 nanometer. Zeta potential we found to be negative, so these are the negatively charged nanocarriers. We also calculated the drug entrapment, which is found to be more than 90%, and the drug content was about 4% weight by weight. So after initial characterization in terms of the size, charge, and the entrapment, we wanted to see if the developed nanocarriers were able to increase the lung residence. I mean, the residence was able to control the release of the drug from nanocarriers. As you can see, in this graph, the percentage dose release versus time. So this is the graph, a drug release if you have a solution. And then this is the release profile for our nanocarriers. So, these nanocarriers, indeed showed and control release property, till 76 hours when they are basically, developed as a nanocarrier system. So for this release studies, we use a simple setup. We took a dialysis bag, put it in a basket of USP type 2, and took the medium of 200 ml, and conducted resolution, took the samples at specific time point, and determined the drug content. So, with, as we know, our drug, the drug is getting released in a controlled fashion, from the nanocarriers. The next question is whether, these nanocarriers are suitable for inhalation delivery or not. So for that one, we use a USP recommended 8-stage cascade impactor. This is the Anderson cascade impactor. It basically mimics, lung. As you can see, this is an induction port where it mimics like a bronchi, and this is a bronchi mimicking bronchi. And we have 8 different stages, and each stage has a plate. So it has a plate which is of specific particle size cutoff. So what that does is, once we nebulize, the formulation with the help of compressor, here we have nebulizer, the larger particle size or particle droplets will deposit in the upper plates, and then the smaller particle size droplet deposit in the lower plates. And by using this data, we can determine what is the fraction of the drug which is reaching into the deep lung. So definitely, once you have a setup, you put the formulation, you have nebulizer, turn it on, we turn out the vacuum so that the air is sucked from the nebulizer, and the aerosol cloud will be deposited in each plates, and we took out those plates and determine the drug content. So what we found is, mean mass aerodynamic diameter, it is the diameter of droplet which reaches the deep lung, we found to have 1.58 micron. Geometric standard deviation talks about the overall particle size distribution. We found that it has anything less than 2 is ideal. So we found 1.26 micron geometric standard deviation. Fine particle fraction, it is the amount of the drug which is less than 4.7 micron. And then we found on 76% of the drug is releasing into the expected to reach the deep lung. You may ask a question that, well, the MMAD is 1.58 micron, then how the nanoparticles are getting deposited in the deep lung? So because anything less than 1 micron is expected to aerosolize, expected to exhale, once we breathe out. So basically, in this case, what happens, we have a nanocarrier suspension, then it generates the droplets containing multiple nanoparticles. So it is a microparticle droplet comprised of multiple nanoparticles, and that's how it reaches drug to the deep lung. So after that, next one, we wanted to see if during the aerosolization process whether these nanoparticles are stable. So for that purpose, we use a viable cascade impactor. So this is the viable cascade impactor where you can put the petri dishes in each stages, and then we nebulize the formulation, and then took out the petri dishes and determine the particle size and drug loading. So we found that during the nebulization, there was a non-significant change in the particle size and drug loading. So now we know that the nanocarriers were able to control the release, as well as they are suitable for the inhalation delivery, and they are stable during the aerosolization. So we built a rationale, basically to further continue or continue to explore the use of these nanocarriers. So what we did, we did a lung deposition studies. We used a similar setup as I described earlier. So in this case, we took the biopsy mice, and one group we had a aerosolized silica solution at a 4.82 meg per kg dose, and then we had aerosolized silica nanocarriers at 1.47 meg per kg dose. If you see here, there is a threefold lower dose with the nanocarriers compared to drug solution. We nebulized for 30 minutes, and then we took the plasma, took the blood and lung samples, lung tissue from 0 to 24 hours, and then determined the drug content in the plasma and the lung tissues. So if you look at the percentage dose of silicazib deposited per lung tissue over the time, indeed, silicazib NLC is able to increase the lung residence time. So this is the profile of an nanocarrier system, and this is the drug solution. As we know, most of the drug get cleared in 2 hours. However, in case of nanocarriers, we did found significant higher drug concentration even after 12 hours in the lungs. We found in a similar findings in the plasma. This drug is also getting absorbed in the plasma. And we determined the pharmacokinetic parameters, and important to highlight here, we found that the nanocarrier system provided the area under curve of 1.26, and if you take a solution, it provides 1 point almost 4 or 1.36. So as you can see, by using threefold lower dose, we are able to fourfold increase the area under curve. So that basically supports our hypothesis and confirms that the use of nanocarriers or the developed nanocarriers were able to increase the lung residence time of the drug, and they are suitable to deliver the drug to the deep lung. So the next one, what we did, we used our orthotopic tumor model where we took 1 million A549 cells injected into the lung parenchyma, and then waited for 7 days, and we randomized mice into different groups. So in this case, we had a control group treated with a placebo aerosol. Placebo means there is no drug in nanocarriers. Then we had a aerosolized silicazib solution. We had aerosolized silicazib nanocarriers. Once again, you see we have threefold lower dose with nanocarriers compared to drug solution. And we use intravenously administered docetaxel, and then we combine this intravenously administered docetaxel with aerosolized silicazib solution and the nanocarriers at respective doses. We continued this treatment for 32 days, and then at the end, we sacrificed the mice and we removed the lungs and measured the tumor volume. So if you look at the tumor volume data with different control treatments, indeed, the silicazib nanocarriers did show a significant anti-cancer activity compared to control. Again, we are confirming here silicazib solution alone didn't show a significant anti-cancer activity, but the nanocarriers were able to increase were able to show a significant anti-cancer effect, supporting our hypothesis. And when we combine with the docetaxel, this combination of nanocarriers, found to be significantly different than even the single agent treatment. So even it is outperformed the silicazib solution combination. So with that, basically we have a nanocarrier system which is very promising. This is in terms of the efficacy data. We also wanted to check whether placebo nanocarriers are toxic to the lung, because the excipient, anything you put in the lung, lung is very delicate and we need to test that. So what we did, we evaluated the toxicological profile of placebo nanocarriers as well as drug-loaded nanocarriers in the biopsy mice. We aerosolized, the formulation for 25 days by using our in-exposed nose-only exposure chamber, and then determine the acute lung injury assay by measuring wet to dry lung wet ratio. And we found that placebo aerosol as well as lung aerosol wet to dry lung wet ratio, which is falling into the normal range. We also look at the myeloperoxidase activity assay, which gives information regarding the function of leukocytes, and our formulations were basically not having any impact on the leukocytes. So with that, to summarize, we have a delivery system. We're able to increase the lung residence time of silicazib. We are able to achieve the significant anti-cancer effect by from aerosolized silicazib nanocarriers alone as well as in combination, and it has a safety profile. So we can use these nanocarrier systems for the inhalation delivery in order to enhance the anti-cancer effect and also minimize the side effects. In this investigation, we did it for lung cancer. We are also exploring these delivery systems for asthma, as well as we have one project started on COVID-19 recently, where we wanted to see the efficacy of these lipid particles to deliver the drug in the lungs. So with that, that was basically the research work I wanted to share with you all. Um, um, you know, there are different types of therapeutic agents. So you may have the small molecular inhibitors. You may have proteins and plasmids. You may have the sRNA and gene delivery. So not all therapeutic drugs need nanocarry systems. We need to really see what are the best candidates, have a good significant anti-cancer activity and suitable carriers or suitable drugs to develop in a nanomedicine. So for example, these are some small molecule. We have this is STAT3 inhibitor, and this is a DFMO we have. This is anti-cancer drug, but they have a short half-life. So we can increase the half-life of the drug using nanocarriers. With the sRNA, you may know it is very challenging to deliver the sRNA across the cell membrane. So we need a nanomedicine system for the sRNA delivery. We also work on the protein delivery. As you know, it's very challenging to deliver the protein across the membrane. So, these are the basically therapeutic agents, we are working on. And as I said, we really see, or we really screen the rationale behind, development of the formulation or nanocarry system. So in other project, we develop a hybrid nanocarry system. As you can see here, the inner core is albumin nanoparticles and the outer core is chitosan polymer. So, and then, we have a pegylated antibody on the surface to specifically recognize the antigen, which is overexpressed on the tumor cell. So the advantage of, this hybrid system is we can either load two drugs in albumin nanocarrier or core particle, or we can we can load one drug in albumin nanocarriers and other drug in the chitosan outer polymer. Um, and the pegylation again, to enhance the plasma life. These are intended for intravenous delivery. We are also working with the bioengineered polymeric nanoparticles. Those are synthesized by chemical engineers, and we explore the natural polymers such as albumin and chitosan. We are also working on the liposomal formulations for oral as well as inhalation delivery. And with the route of administration, currently we are focusing on the parenteral and the inhalation route. We did some work with the oral, but that is we are not focusing on the oral route of administration at this time. So with current focus in my research lab, we are focusing on natural polymer or lipid-based, drug and gene delivery system. The other focus is inhalable delivery system, and then the third focus is, have developed a targeted combination therapies, so that one of the advantage with combination therapy is we are able to use the lower dose of the drug, achieve significant anti-cancer effect with the lower side effects. So that's where the rationale comes in for the combination therapy for any disease, specifically with the cancer. Due to the heterogeneity, we have to use the combination therapies. With that, I would like to thank NHLBI, that's the National Heart, Lung, and Blood Institute, for awarding an NIH R15 grant, is a 3-year grant. Also, I would like to thank National Institute of General Medical Sciences for Research Continuous Award. This was my first NIH grant. I did got multiple grants from Hawaii Community Foundation. Yeah, I would like to thank these funders that definitely helps to advance, the research, and the ultimately goal is to develop the formulation eventually will be used in the clinical studies. With my collaborators, Dr. James Truxon, he's from University of Hawaii Cancer Center. So his expertise is um, uh, medicinal chemist. So he develops new compounds which we are testing. Stephen Seb at Texas A&M University, and Mandip Singh was my postdoc advisor at Florida A&M. And we are also worked on the bioengineered polymer, which we got it from Dr. Sung Wang Kim from University of Utah. You may know, Journal of Control Release, which is basically Dr. Sung Wang Kim is one of the founder of that journal. Currently, I have two PhD students, Prasad and Neeraja, and a master student, Ray, and I have a postdoc who recently joined. I have alumni, from my lab, starting from the postdocs, graduate students, as well as the international visiting scholars. With that, I would like to thank you, all of you, for your valuable time. And I also thank, Dr. Chandivar sir for giving this opportunity, as well as Dr. Gaunde madam for organizing this seminar. Yeah, so with that, I will be happy to answer any questions you may have.
Thank you, Sogule sir. Hello.
Yeah.
Yeah, am I audible?
Yes, yeah, you're audible.
Yes, thank
And
you so
we can
thank
take
you,
the
Sogule
questions,
sir.
yeah, from
Now I
audience.
request participants to raise the hand for the question answer session, and I hand over this question answer session to Kitukule ma'am. Over to you, ma'am.
Thank you, ma'am. Thank you. If you have any questions, you can raise your hand.
We can also take the questions, by chat. I mean, if someone wanted to, write, that is also fine.
Also,
You can
Girish
accept
Darvekar
chat.
sir, who is on YouTube. Participants who are on YouTube, they can Girish Darvekar sir will ask the questions.
Hello, Girish. Is there any question from YouTube?
Hello.
Yeah.
Hi.
Still, there are no questions on YouTube, sir.
Okay.
Sogule sir, there are certain questions in the chat.
Yeah, sure. Yeah. Yeah, so I see one question from Shweta Saiker. Could you please tell us about the combination therapy of chemo drugs? So, there are a lot of combination therapies currently being explored. Whether you have a combination of two, chemotherapeutic drugs, or combination of immunotherapy, or in this case antibodies, along with combination of the drugs. So if you look for the lung cancer, we have for the lung cancer, we have different small molecules. Those are given by oral route of administration. Just a minute, let me see what's going on here. Yeah. So yes, we have some drugs, those can be given orally. That's one of the most suitable dosage form. And then we have the parenteral drugs, those are in the combination. In terms of the inhalation delivery of chemo drugs, it is suggested to be an adjuvant therapy, which is under development. It is not yet approved by the FDA. Yeah, let me see the next question. Yeah, the next question is from Chandan Tiwari, that which method suppresses the lung cancer fast and effectively? So one thing, nowadays we have to profile the lung cancer, lung tumor. What doctor does is, they take the biopsy of the sample, biopsy of the tumor, and do a gene profiling to see what are the proteins are overexpressed or underexpressed. And based on that profile, the targeted therapy is recommended. For example, if you have an EGFR overexpressed lung cancer, in that case, the EGFR inhibitors are given. So that's just to give an example. So those kind of targeted therapies, can be given to the patient. Again, it depends on individual patient and the makeup of the tumor. Doctor definitely goes with the first line treatment, second line treatment, and third line treatment. The first line treatment, where is firstly tried with the drugs, and doctor basically titrates, these drugs. He will try, one after another, depending on how patient is responding. Doctor also takes into the consideration of the side effects, because one drug may be very effective, but if it has a side effect and patient is not able to tolerate the drug, it's gonna to basically hamper the health of the patient. So in that case, um, uh, in that case, the doctor is not able to continue that specific drug, then he tries an alternative drug which may have a different side effect or a different mode of the action. Yes, so we are getting more questions. That's great. How to measure a tumor volume? Which apparatus is used for it? So with the tumor models, most commonly used, there are two types of tumor models. One is the xenograft model. What is meant by xenograft model is basically researchers inject the tumor cells in the flank, left or right flank of mice. So it is a xenograft. It is a tumor grown other than where it is in, grows in the patients. That's a xenograft model. So in that case, how to measure the tumor volume? Use a vernier caliper to measure the length and diameter, and from that we'll get the tumor volume. At each specific time point, you measure the tumor volume with the vernier caliper. In case of the other model, which is the orthotopic tumor model, what does that mean is the tumor is developed within the lungs, so that is more relevant to the clinic. So with that, orthotopic tumor model, one option you have is at the end of the study, you basically harvest the lung and then isolate the tumor nodules from the lung, and then you measure them, and that's how you get a tumor volume data. So if you wanted to have a ongoing or live, progression results for the tumor, in that case, you need to use luciferase expressing cells. So, then with these cells, you can inject the luciferin at each time point. You can see the bioluminescent signal, which will tell us that, after 1 week, what's happening, second week, third week, fourth week, and so on.
Thank you, sir. And I think Miss Priya Munshi has some question. So I have unmuted you, Priya. You can ask your question.
Okay, it was really informative session, sir. I just want to ask you one thing. Like, isn't genomic testing is an option for lung cancer? Will you explain it?
Yeah, with the genomic testing, what they do, they take the biopsy of the tumor and then do the gene profiling. So they will look at different genes, for example, EGFR gene, even the mutated EGFR. Then we have a BRAF gene, then we have a RAS, KRAS. These are some examples of the proteins. They look at the expression of protein, and they have the control, which will be the normal lung, and they will find what are the proteins are overexpressed in the tumor. Those are driving the tumor growth. So if say, like, for example, you have EGFR which is overexpressed in the tumor, and if you use the EGFR inhibitor, that's basically the first line treatment doctor will start. um, um, and then like, for example, KRAS, if that is a mutation there, then there will be drugs. Those are specifically works or inhibits the KRAS protein. Then with that inhibition of protein, we are able to reduce the lung tumor or shrink the tumor size of the patient. So yeah, this is commonly used nowadays. Even in India, I know some friends. This is also commonly done, testing to see, what kind of genetic profile specific lung cancer has, and based on that, then doctor tell us the the treatment. So which is actually a very useful tool, because before giving a drug, patient is likely to respond to specific drug. Not like you give the drug and then you look like a patient how is responding. So it is very proactive approach, and very good approach to do the profiling of the tumor, and based on that have a different therapeutic options for different disease. And again, as I said, as you see, one combination may be working for one patient, but that same combination not necessary it will work for other patients. So we have to have, and that's where the personalized medicine approach or strategy comes in. So we are developing the treatments in a personalized way, so you have specific treatment for specific patient.
Thank you so much, sir. Satisfied.
Yeah, no
Okay.
problem.
There is one more question in the chat box, sir, from Hyundai, sir.
Yeah, there is one more question I have. See, okay, just so can we minimize the side effects of conventional delivery system by this targeted drug delivery system? So short answer is yes, we can do that, and which actually we in our lab did in another project where we developed this nanostructured lipid nanocarriers. And what we did, we used the DoxNT as a linker and then attach a targeting peptide on the surface of nanocarrier. And this targeting peptide basically recognize the tumor cells, not the normal cells, or very minimum, normal cells. So by doing so, we can, it's called as active targeting, you may be familiar with. So by doing so, we are increasing the drug concentration or the concentration of nanomedicine or nanoparticle at the tumor site while sparing, the normal tissues and organs. So yes, we can develop it, and as I said, you can have antibody on the surface of the nanocarriers, or you can have a peptide which basically binds to specific receptor, and then we get a receptor mediated endocytosis within the cells.
Hello.
So there is
Sir,
another
yeah,
question.
sir, I have also, sir, actually, I have also one question that it is very basic question, sir. If we want to develop our lab for the anti-cancer activity, what are the basic needs of equipments or facilities that are required?
Yeah, so for the anti-cancer research lab, right? So there are two, I will say, two research labs. One is basic cancer biology and therapeutics lab, where you need a facility to grow the cells. So you need a cell culture facility so that you can test, the effect of these different drugs or even the nanoparticles, in vitro before going to the in vivo animal models. The second facility you will need is the animal facility, so that you can conduct, this orthotopic or xenograft tumor model, as well as do the pharmacokinetic studies to understand absorption, distribution, metabolism, and excretion of specific drug or even the nanomedicine. The second type of lab will be like a drug delivery system or nanotechnology based lab. In that case, you will need the cell culture facility as well as animal facility. In addition to that, you will need the particle site characterization equipments like particle size analyzer, or zeta potential measurement. So at least you will need that basic equipment to conduct the research in that direction. Yeah, with the research lab, I would like to just share my experience. So when I joined University of Hawaii, so again, basically I got an opportunity to design my lab and get all the equipments and establish the lab. Same thing, once we moved to Mississippi, also we expanded our lab. But feel free to contact me if you need any specific question, or if you need any specific information, I'll be happy to help. If you'd like to pursue in that direction.
Okay, thank you, sir. We will need your cooperation, sir.
Yeah,
I think Dr. Samir Lakde is also having some question. I have unmuted you, sir. Please unmute yourself.
Good morning, sir. This is hello.
Yeah, good
Hello,
morning.
good
Yeah.
morning, sir. This is Dr. Samir from Pune. Yes, sir. Sir, my one question is the fundamental properties of anti-cancer drugs such as LogP and pKa values are important
Yeah.
for designing the delivery methods as well as the clearance from the lungs. So what will be the criteria for anti-cancer drugs like pK value and LogP?
Yeah, with the log P, log P we calculate based on the external water coefficient studies. Um, so if you have a higher log P, which tells us about compound is highly lipophilic, and if it is a lower log P, it tells the compound is hydrophilic in nature. So in order to
Yeah.
for the delivery to the lungs, definitely you will need a hydrophilic and lipophilic balance, because the systemic circulation is aqueous, right? And then from there it needs to go within the cells, and cell membrane it has both aqueous component as well as the lipidic bilayer. So it needs definitely a combination of um, a proper balance of hydrophilic and lipophilic properties. Again, it depends on drug to drug, so we need to uh, decide case by case basis. The answering your second question regarding the pKa, yes. So with the pKa, one of the thing, like you can take an advantage of pKa where drug is dissociated and have a positive charge under the physiological condition, where it can interact more with the cell surface, which is a negative charge, and we can enhance the delivery of the nanocarrier system. So in case of liposomes, I can tell you by parenteral route, what is found, either neutral or negatively charged nanocarriers are helpful, or they're able to target higher fraction of the drug to the lungs, compared to like positively charged. Yeah, I hope that's answered your question.
Hello.
One more question is from Shweta Saikar in the chat box.
Yeah. The question is, anti-cancer drugs are being used for the treatment of COVID-19. Is it safe, and what is the logic behind using it? Because anti-cancer drugs will definitely have a side effects. So, not, I mean, again, it all depends on the drug, whether it has a severe side effects or it has a minimum side effects. Just to give an example of celecoxib, right? So if you take it even by orally, if you take it even in grams, you're not going to see severe side effects. So those kind of drugs can be, repurposed for COVID-19. But specifically the drug, those are antiviral in nature. We use it, but in any therapy, we have to look at the risk to benefit ratio. So if risk to benefit ratio is pretty good, so in that case, it is worthwhile to test that specific compound, or use that specific compound in the patient. So like in case of COVID-19, one of the major issue is the mortality. So any drug which is reducing the mortality, or even the drug which will prevent the overcome the rate of mortality, will be a blockbuster in case of COVID-19 at this condition, before the vaccine is developed. Even with the vaccines, there are different types of vaccine are under development for COVID-19. They have different either based on DNA, or based on the virus particle, or based on other virus particles. Again, everyone has a different platform. I think in India also there are a couple of companies developing the vaccine. Um, Depending on its efficacy, we have a long time to go. But those kind of strategies can be used. And one of the things why you want to repurpose the drug, because we know properties of the drug, safety and efficacy of the drug, so that it will reduce the path to reach the clinic, so that they can start clinical trials, and then followed by FDA approval.
Is there any question in chat box, ma'am?
Sir, one more question from the Channavar ma'am.
Yeah. So yeah, tell us about type of natural lipids suitable for formulation of nanocarrier systems. So one of the natural lipid is DPPC. So it's a dipalmitoyl phosphatidylcholine, which is available, in our cell bilayer. Also, it is available in the lungs. So you can use DPPC plus cholesterol. We all, all cells have the cholesterol. That helps to increase the integrity of the bilayer and reduce the drug leakage. So those, I think, two lipids can be used. Even HSPC, hydrogenated phosphatidylcholine, can be used. I think compared to DPPC, I guess HSPC is cheaper, but yeah, that is also very safe, to use.
Sir, I have one question.
Yeah.
What is to which extent this lung cancer patient can be treated?
Actually, with some of the treatments, we see, the patient is, tumor free. But as I said, depending on the type of the tumor and its genetic profile, how aggressive it is, and what stage it is diagnosed. That's another important aspect, the diagnosis. If it is early diagnosed, and then you have better chance of patient getting recovered from the tumor. But if it is diagnosed at the later stage, and if it occurs the metastasis, then it's very, very challenging and almost difficult to treat the metastatic tumors. But as I said, with all different drugs and treatments are available, we are able to, in some cases, get a tumor free survival of the patients, and some cases, they are in between, and some extreme. But if you look at the overall, survival rate is basically 16%. So you can get an information regarding the success of these different therapeutic approaches.
So
Thank
one
you,
more
sir.
question in the chat box, I think from Ms. Pratiksha.
Pratiksha Lambade, YouTube.
Yeah, YouTube.
Yeah, I think I'm not able to see the questions from YouTube.
From YouTube. Sir, which type of treatment for lung cancer are permanently cured? This is from YouTube viewer.
Yeah, so actually there is no permanent cure for lung cancer. That's the unfortunate thing. So, you will see the treatment, you can reduce the treatment, but there is no medicine so far. Even people are developing vaccine, but yeah, we don't have a cure, for lung cancer. Or even most of the cancers, it's very difficult to find a treatment for this. That will be ideal, but we don't have any treatment which cures the cancer.
Okay. I think most of the questions have been resolved.
Yeah.
Okay. Thank you, sir. Thank you for
Yes.
your generous answers. And it was very nice and informative
I
session.
think one more question, Samir Lamade, chat box. You check the chat box.
Yeah. What
It
are
is
the
already resolved.
what are the underlying complexity in chemical and physicochemical phenomena has been noted, I guess, and if so, how to overcome it? Chemical and physicochemical phenomena. So, the chemical and physicochemical properties of the drug can be modified. For example, you can prepare say, like, if you wanted to enhance the solubility, you can prepare a salt of a drug if it is a weakly basic or weakly acidic. So you can prepare a salt, it will help to increase the uh, solubility of the drug, or the rate of solubilization of the drug, or rate of dissolution. Like if you wanted to again enhance the oral viability, you can have a drug in amorphous state compared to crystalline state, which will have higher solubility in the amorphous state. And on the chemical side, there are reports where they're developing the prodrugs. That is another area of research. There is also drug antibody conjugates. Those are nowadays, another therapeutic category widely explored for the treatment of cancer. Yes, it is it is possible to modify and tailor those chemical as well as physical chemical properties in order to achieve a desired effect, whether it's drug absorption or if it is an drug delivery at specific tissues.
Ma'am. Is there any more question?
I think one more question from Samir Lakde, Dr. Samir Lakde.
Yeah, the thing is says the underline noted here. How to overcome it, like self-association or molecular aggregation. Okay, so self-association and molecular aggregation, if you wanted to remove, I mean, you wanted to prevent, um, I say how to overcome it, like self-dissociation or molecular. I guess, I see it in a two way. One way is we wanted to prevent the molecular aggregation and self-association, which can be done with the help of the polymers. You can have a drug polymer mixture, and even in different delivery systems to overcome that. And the other side, if you wanted to have self-association and molecular aggregation, then you have to use specific agents like surfactants. They help either way to enhance, prevent the molecular aggregation, and some will enhance the molecular aggregation based on the property of drug as well as the property of surfactant used in that case. Yeah, so the lot of work has been now going on in terms of the molecular amorphous dispersion. So we have some products on the market also, so where you have a drug dispersed in a polymer, which is an amorphous in nature, highly soluble. It increases the oral bioavailability compared to a crystalline drug. Yeah. So we have, I think, another question: how much time it takes for medicine to impact on cancer tumor? So, the medicine will start working on the tumor cells, I would say within 12 hours. So initially it will take some time to achieve the therapeutic concentration. So within 12 hours, it will start showing the effect. But I think in order to see the time point or significant anti-cancer effect, you need in a couple of weeks of the treatment. That's why you will see with the chemotherapy, there are rounds of chemotherapy. They give like, okay, six chemotherapies, and then 8 chemotherapies. So these kind of different rounds of chemotherapy is given, and it generally, I will say, a few weeks it will take to show the effect.
Thank you, sir. Now it's over to Gawande ma'am.
Thank you, Sobule sir, for very informative session. It was very nice session, sir. And I request Dr. Pranita Kashyap ma'am to express her views as she is a chairperson of today's session.
Thank you very much. Can you hear me, all of you?
Yes, yes, ma'am.
Hello, Sobule sir.
Yeah.
Very nice presentation was there, and thank Tandevar sir for giving me this opportunity to chair this session. And now sir has told most of the things about the cancer, how he is working. He has answered all the queries regarding cancer and development of lab and everything. And he's so kind and ask ma'am to at any time he can be contacted to develop the lab. Sir, sir has given the targeted drug delivery system. As all of you know that cancer is, we fear, first fear was the cancer, and second now it is COVID. So he's working on such a important, if not it is important, but in the treatment it is very important, that is the cancer. And in the cancer, most of the time the toxicity of the drug is very much concerned, and that is why his work is on the targeted drug delivery and nanoparticles, so which reduces the toxicity, also residence time in the lung. So he is doing a great job. And now his new project on the COVID. So I wish you best of luck for this project, sir. And you are doing a great job for the humankind. And also that is the extension of the Indian for the contribution of this session. So I congratulate you, sir. Very nice session, sir. Thank you very much. Thank you.
Yeah, thank you very much for kind words. Yeah, I appreciate it.
Thank you, ma'am. Thank you very much.
Thank you.
Now, for the vote of thanks, I would like to call Miss Nilima Kinekar.
Thank you so
Over
much,
to you,
ma'am.
Nilima.
Thank you so much, ma'am. Am I audible, ma'am?
Yes, yes.
Thank you, ma'am. I, Ms. Nilima Kinekar, Assistant Professor, Pre-Vadwani College of Pharmacy, Yavatmal. I take this privilege to propose a vote of thanks on behalf of management, Principal, Professor Dr. A.V. Chandevar, sir, Dr. Nitin Kochar, sir, Session Coordinator of this international webinar, Dr. Abhijit Srirao, sir, Associate Coordinator, Dr. S.R. Gavande, madam, Session Coordinator, and Ms. Kitukle, madam, also Session Coordinator, and entire team of Vadwanis. I thanks Dr. Mahavir Chogule, sir, who is Associate Professor, University of Mississippi, for their valuable time and enormous guidance on today's topic. Thank you so much, sir. Once again, thank you, sir. Also, I am very much thankful to Principal Professor Dr. A.V. Chandevar, sir, for their continuous support and guidance. I also extend my gratitude of thanks to Mr. Raulin Singh Khurana, sir, for wonderful inaugural speech on this international webinar on pharmacy 360-degree research to practice. Also, I thank to today's session chairperson, Dr. Kanita Kashyap, ma'am, principal at Dr. R.G. Goyal College of Pharmacy, Varda. Thank you so much, ma'am. Also, I want to thank Dr. Nitin Kochar, sir, who is the professor of Department of Pharmacology and coordinator of this international webinar. Dr. Abhijit Rao, sir, assistant professor, Department of Pharmacology, who is associate coordinator. Also, I want to thank Dr. S.R. Gavande, ma'am, assistant professor and coordinator of today's session. Ms. M.D. Kitukle, ma'am, principal of D Pharmacy and today's session coordinator. Last but not least, I want to thanks to all the PWCOP teams for their continuous support. Also, I want to thank all the participants who have actually participated in today's webinar. Here, I concluded that today's session is over. We will meet tomorrow at same time at 4 p.m. with same link, same ID and same password. Once again, thank you. Thank you so much, sir.
Hello, Mahavir.
Yeah.
Thank you very much.
Yeah, no problem. Thank you. Thank you
Yeah.
for the opportunity, and I wish great success to this webinar series, and we'll be happy to also get involved in the future webinars.
Thank you, and we're proud of you that we are doing wonderful job. Your research use anti-cancer drugs, which is a need of today's era. So keep it up, and thanks again. And be in touch whenever you will come back to India. Call me.
Sure.
Right, we will meet personally. I will call you to my college, to your college, rather than. Thank
Our
you.
college.
Yeah. Thank you.
Yeah, thank you. Thank you very much.
Thank you, Dr. Mahavir. Nice to
Yeah,
see you after a long time.
Yeah, thank you, madam. Yeah, it's good to see you. How are you? How's things are
That's
going
fine,
well?
very fine, very fine.
Oh, great. Yeah, good. Yeah, I saw. I mean, Facebook. I see some links or
Yeah.
some news. Yeah, but it's good to see, good to meet you via Zoom after a long time. Yeah, I think it's almost maybe 13 years now, right? Not more than that, I guess. Yeah, maybe 15, more than 15 years.
Okay.
Thank you, Dr. Mahavir.
Thank you.
Kashyap, madam, for being with us.
Yeah, sure. Thank you very much, and have a good day.